tutorial questions, and if you have any questions, you can ask me. What I want to discuss, the same thing what I presented in the morning and also afternoon. I just give you a glance of all those and little bit here and there. I want to tell you the story of some of these questions, and then I also answer to those questions, like, a story again, you know what I'm saying? Not to seriously work on the questions and answer. At least you don't have to worry too much about it. If you don't understand, it's fine. But if you understood the concept of the talk in the morning, definitely these questions are answerable in your own way. But if you don't have it, it's fine too, because that is the story I wanted to deliver and give you, so that you all understand what it takes a PhD students to, accomplish something, and also establish herself to become a great postdoc in a great university and a mentor, and also learn. Even though she was a cancer biologist and she learned immunology, she passioned immunology, and she started working on immunology, right? It's about that you change. I was a food biochemist and become a cancer biologist, right? You may be someone like can become something else someday. So it's about what you are interested in, and during your process, while studying, understanding what you are working on, probably you'll learn something and you take that and move on, right? Whatever that you are passionate. Okay. So question is, why is targeting cellular signaling critical in developing therapeutic strategies for PDAC and pancreatic cancer? It doesn't matter pancreatic or any other cancer. So if you cellular signaling, right? So it is critical, because doesn't matter which cancer you want to treat, all these cellular signaling pathways are the one actually, helping all these, other signaling in other populations, right? Either tumor intrinsic or tumor extrinsic, they all regulate through cellular signaling. Even though you take the tumor extrinsic other cell population, it's also in the air. There is another signaling pathways that regulate. So targeting these pathways, well known in the field many years, that targeting some of these factors. For example, you say one cytokine like IL-1, you can actually block IL-1 too. Okay, like that you can actually block some cytokine. He was talking about IF-1 alpha. You can block IF-1 alpha, and actually you study and you get the same kind of targeted therapeutic. But signaling is important. Okay, you have to understand the mechanism. So that's why signaling is important. It doesn't matter what you treat, but as a biologist, you need to tell the story of how did you do, why did you do, and all those things, right? The next is, what role does P38 MAP kinase play in pancreatic cancer, particularly in relation to IL-1 alpha? See, the P38 MAP kinase is overexpressed in KRAS mediated pancreatic cancer, and also it actually regulates, I presented that data, regulates IL-1, phosphorylates a lot of other SP1, other P65, that's a NF-kappa B factors that led to IL-1, a lot of IL-1 production. This activates on inflammatory CAF, and also it supports the immunosuppressive microenvironment. So those answers that I was giving you is all derived by experimentally conducted experiments and kind of worked on and showing that this has happened. So you basically conduct experiments to prove something, right? To prove something that you have a question. So that's exactly what happened, the overall, right? So there's a question, how does IL-1 contribute to the tumor microenvironment? I don't want to say IL-1 only. Any cytokines you take, chemokine you take, or any factors that you take released from the cell. See, there are many factors released from the cell. Okay, they are all in the bloodstream too. Okay, there are many things released in the bloodstream. Blood is not blood. It has everything you think of, because it is flowing, going from one body part to another body part in the whole body. That's why blood has a lot of chemical in it. Okay, so in that we were just taking example of one IL-1, Like that, there are so many factors that are running around. Okay, they go one body to another body, one organ to another organ. So, for example, let me tell you, if IL-1 is bad, okay, the bad IL-1 from a pancreas, okay, is released, and if it is really bad, it is actually flowing to other organs too. But it's only the factor is running around. If the factor is going and running around, it is not make the metastasis. Your cell has to go, then only it will be metastatic. Okay, your factors may be in your body, bad one, good one, are all there running around, but they are okay. Okay, there is nothing wrong. But that's how the bloodstream work. They go to heart also, they go lung, they go everywhere. But the bad part is that your cancer cell, which actually metastasized to another organ, is a bad thing. You have to control that cancer cells to go out of your cell type. That's why if you can reverse, like for example, I always say, in aging, I wanted to reverse the aging. The same way, I want to reverse the cell shape towards the normal, how much ever I can. Every day I think about it, like, how do I do it? Probably one day we may do it, but what I'm saying is every day we think about that one issue that I had it like in 25 years ago. I keep thinking, how do I reverse it? What actually makes? You keep reading and probably one day you will figure it out. Probably not. It's okay, but you get the knowledge. At least I've read a lot of papers. You know what I'm saying? So like that, the factors, we are only giving you an example. But you have to understand from that one example, like so many things that you can actually study, like, understand also, okay? What experimental evidence supports the use of P38 inhibitors in reducing IL-1 expression? See, we have showed the preclinical studies, pharmacological inhibitor like, MAP kinase inhibitor, and also we studied with the knockout, knocking out the P38 is important. You don't do just only the chemical. You have to do with the knockouts and gene regulation, knocking down and all those experiments, and prove your hypothesis in genetically and also pharmacologically always. Okay, which transcription factors are implicated in the P38 MAP mediated regulation IL-1, and how are they affected by P38 inhibition? The transcription factor I already told you, SP1, NF-kappa B. Which are critical for IL-1 transcription. The P38 MAP kinase inhibition reduced this phosphorylation, nuclear localization. You saw all those things, and also it led to a reduce the IL-1 promoter, also IL-1 expression. So what I'm saying is that when you study, something pop up and in your study, and you need to understand either cellular intrinsic, extrinsic, and also nuclear, cytoplasmic, and if it is nuclear, what it is, and cytoplasmic, what it is, and whatever the molecule you are studying, whether it is a cytoplasmic, it's related, or the nuclear, you have to work on and study. You have to experimentally prove that though, cytoplasmic and also nuclear, then divide your experiment and conduct the way she conducted Samara, like herself. Like, then she did the inhibitor study, and also she did the knockout study. These kind of things probably give you a lot of answer. So how does the inhibition of P38 affect the CAF activation in cancer? So by reducing this IL-1 alpha P38 inhibition, it actually, you saw, it actually impairs the inflammatory activation of the CAFs. So also you saw how this growth factor cytokines, pro-inflammatory cytokines, and growth factors of CAPS, which can also reduce the desmoplastic reaction. She actually showed some fibroblastic reducing the fibroblast cells. Like in IHC, she did staining the trichrome blue and Sirius red and all those things, and she showed like that. In each cancer, it's important is that, what is that important in each cancer? That in our pancreatic cancer, stroma is important to study. So that's why she did that. In other cancer, there may be something else. You have to do those and conduct those experiments, right? So what are the main differences between IL-1 alpha, beta? You already know. IL-1 alpha, tumor intrinsic, epithelial, you already saw the data and everything. And she did IL-1 beta in the other population, macrophage population. She saw IL-1 beta was there. And then she inhibited the P38 in IL-1 in those population, and she saw that IL-1 beta went down. So basically, you saw the difference that she did not do IL-1 beta in the tumor intrinsic. She only did with the other population. So like that, if someone has published some paper on, okay, what are these do, what are these going to do, that is true. What she saw, what others have published in paper is also the same thing. So she just proved it in our paper too. That's important. Proving the idea and the concept ideas from others, again in your work, is the important thing. So you have to prove it and keep going, proving it, and also proving the hypothesis that you generated, right? So describe the impact of P38 inhibition on the tumor microenvironment. So it's just when you inhibit P38, it's just not only reducing the inflammatory CAF activation, right? It also remodels the immune components. She studied each population. She studied like macrophages, monocytes, MDSCs and T cells component. She saw all the T cells, CD4, CD8 went up. You know what I'm saying? So like that, you have to study different cell component and do. That's the beauty of the technology that we have today on single cell sequencing. So how does combining MAP kinase inhibitor and chemotherapy improve the therapeutic outcome in PDAC? Because the chemotherapy is a standard therapy already improving in the patient, right? So now you add with the synergistic combination of this P38. So it did work. It did increase the survival. It is not very great, but I think it did good job. Like at least 2 to 3 weeks is a huge thing in pancreatic cancer. So that's a good sign. So what experimental methods are used to assess the effects of P38 inhibition on IL-1 transcription? If I ask you, you might know already, you might not know. There is a lot of chromatin immunoprecipitation, CHIP assay she conducted to measure the transcription factor, like binding to IL-1 promoter. That's exactly what she did. You have to do the same way. You have to conduct those kind of experiments to prove your scientific investigation. If you say something is regulated, that means that you prove it. Okay, you cannot just say, oh, it's regulated, I move on. No, you have to prove it. So that's what she did. So she proved it herself again and again, right? So it's a key thing. What is the significance of IL-1 receptor localization in calves? You already told you, IL-1 receptor only present. She showed you in all the single cell sequencing data, it was present only on the calves, nowhere else. So that's why she was very confident that her IL-1 alpha has to go and bind on the calves. So because the receptor is right there. So she showed that IL-1 receptor is expressed there. So things like that. So next question is what ways does P38 MAP kinase inhibition alter the inflammatory and stromal phenotype? So the P38 MAP kinase inhibition reduced the inflammatory markers. She showed the IL-6 also. These are inflammatory markers. And CXCL1, I told you we had a paper in the cancer discovery on CXCL1, and it's a great marker too. It's a pro-inflammatory marker. That is also reduced. And she showed a lot of other phosphorylated CAFs because STAT3, because STAT3 is also inflammatory marker, and she showed all those things. So that's a great combination of all these markers, tumor promoting markers. She showed with P38 inhibition. Again, all these markers went down. So next question is how does the CAF specific deletion P38 MAP kinase influence tumor growth in PDAC? So see, the CAF specific deletion in P38 she conducted because the P38 is also expressed in CAF a little bit. That's why she did. And it has been shown reducing the tumor weight and the fibrosis. And the overall, when you do the drug study, when you use the drug, basically the drug is going everywhere, just not the pancreas. It goes every other organ. Okay, every population, like for example, immune population too. That's why the drug can hit everywhere, right? So basically, it did work. It did work because there was P38 expressed in the calf. Probably the drug went and hit the P30, calves also. That's why the overall the therapy worked. Okay. So what future therapeutic implication can be drawn from this targeting P38? See, targeting this P38 it opens up a lot of avenues for us. So we can do like personalized therapy. Particularly with the patient with IL-1 alpha, IL-1 beta. If we can see in the cytokine in the blood sample, if they have a IL-1 alpha, that means that you can expect probably IP38. So you know what I'm saying? Prediction. It's just we don't know. It's just a prediction. So it also provides the strategy to combine with the immunotherapy. We saw that we can actually detect all this in the cells. Some of the cytokines can also activate on the immune cells. So kind of is all prediction. Probably in the future, with the technology changing in technology, we can actually use like personalize something, can do much better work than what we did. So how do the findings on P38 mediated signaling support the design of the combination therapy? See, the highlight that we did on disrupting the key signaling pathway P38 can simultaneously impair this cell-driven inflammation, modulate tumor microenvironment. So, but overall the dual action of this P38 worked when we used the P38 drug because it killed everywhere. So, I mean, basically there are many questions are there. Any research you take, there is a lot of questions arise even after the work is done. Like now, did you take care of the resistance of the mechanism? Did you check the resistance mechanism? Did you check that, this, that? But you cannot answer in one paper, one publication, everything, right? You saw how much work that has been done, and you will not be able to answer all of them. But there is a lot of questions, but we can actually answer one by one later on in the future. But that's how it is. That's how the research is. So, any questions? Anybody?
Sir, did you see any changes in P53 levels after targeting P38 because it activates P53 also? So
Correct, but no, we did not check the P53, right? Yeah, P53. But remember, our model has a P53 mutation already, right? So the one of the model, not the other model without the P53. Now we don't profile the genomic profiling, but when they conducting the patient surgery, after the surgery, if they are treating this drug with to the patient, what they do that after surgery they can get the samples like tissue samples and the blood. They actually take the tissues and also do the genomic analysis. So they do check actually whether what happened. So.
and also, sir, have you checked any upstream genes after targeting P38 and MAP kinase pathway? Any upstream targets have you checked? What is the?
Yeah, you can actually check, but we kind of derived already. So people have already published that it is a KRAS mediated activation of P38. She actually also showed in her paper KRAS mediated activation. Above the KRAS, already we talked about EGFR and SARC and all. There are other signaling comes. So we don't want to dilute the work because it's a lot of work to show everything in one paper. But she actually did all those screening before she went into see the activation of IL-1 regulation and all this. So, any questions? Any general question? Anything about life? Now go back and turn it back because it was fine, we just had to hold it.
One is related to metastasis. Is there any stage, sir, in cancer like metastasis starts? Like we have different stages of cancer. At which stage the metastasis starts? And what makes it to start metastasizing?
So it is not that what stage it starts. It starts after when it is tumor completely. So it has to be a full-grown tumor. The metastatic cells can, after that start metastasizing. And also sometimes in some of the tumors, cancer, we have seen even early on, a little bit earlier than the, during the tumor stage. Okay, for example, in PANIN, we talked about PANIN 1, PANIN 2, PANIN 3. The PANIN 3 is the tumor. So it can start with the PANIN 2 and PANIN 3 any time. These cells can change the shape. And once they change the shape, they start going to the bloodstream. Through the bloodstream, it should be able to enter the bloodstream. So it can be anywhere there. But it cannot be early on because the shell shape is different. So it's hard for them to go and metastasize. Okay, it all depends on the shell shape, right? That only changes during that transition to the cancer. Okay.
And my second question is importance of metabolomics in cancer research.
So importance of metabolomics,
yes,
right?
Like targeting any metabolic pathways will help us to.
Very important, right? See, when you say cancer, the metabolic pathways are high. Sugar, so sugar, they love sugar. The cancer cells love sugar. They want sugar. They want energy. So that's why mitochondria release a lot of energy, a lot of ATP. And glutamate is a bad thing. The high glutamate that is what they want. So if you are giving the cancer cells what they want, I think they're gonna grow like crazy, right? You have to reduce the sugar. You have to reduce the energy from the mitochondria. See what you can do. And also glutamate you have to reduce. And see what you're eating, what you're drinking. That's why doctors say you have to do this. Don't eat that. You know what I'm saying? The reason is that you have to control some of the food that actually giving them the energy, right? So you control those things, and I think can be controlled. So it's about that. So metabolism is a key factor. So any amino acid, all this glutamate go through the metabolic pathway, and energy is through the metabolic pathway. So that the whole metabolism is a highly important pathway to study. Okay, yeah.
Sir, is it possible to stop like metabolic pathway in tumor tissues?
Yeah, there are a lot of studies. Remember, cancer cells are more brainier than the normal cells. Means that they find a way to survive, right? So they find the way to survive. So it's just that. So they find another pathway to survive if you eat with another one pathway. That's how the cancer cells are. But they will always find, there is a lot of targets, there are a lot of studies have been conducted. They are conducting, metabolic pathway is one of the important signaling right now in the cancer field. Thank you. Any question? We have another 5 minutes. You have to speak louder.
Sir, I have a question. I wanted to know how did you get the DRDO fellowship for your PhD?
How many?
How did you get the DRDO fellowship for your PhD studies?
Or DRDO fellowship. So DRDO, they don't have like CSIR at that time. I don't know 25 years or what they have done. I'm telling you the story 25 years before. Okay. So during that time, DRDO announced, mine was the first batch DRDO fellow. Means that was the first year that they announced the DRDO fellowship. I was the first batch student, got a JRF. After that, they keep announcing every year JRF fellowship from DRDO. I don't know what they're doing now, but I'm sure they're doing the DRDO fellowship. That is not like an exam. They actually announce the fellowship. You apply, and they interview only selected candidates in those institutions, And then after the interview, we had a written test, and they also did not know what is GRS fellowship at that time. You know what I'm saying? Because there is a defense, first time students coming, like, they don't know what the DRDO fellowship means. You know what I'm saying? That stage I'm talking about. But we had the entrance exam, and after that, after the entrance exam, we had the interview. Morning entrance exam, afternoon interview. After the interview, they've selected. Only they selected, I think, 4 out of one institution. They only selected 4. So like that. But there was, I don't know now. If anybody has any information, let me know. But I don't know now what they are doing. This is 25 years ago. Maybe 27 years ago. Okay? Good. Okay, thank you so much. If you have any other questions, so we can talk later outside. So thank you so much. I think we had a great day. Hope you all enjoyed it. I think tomorrow is a short day. So, pardon? Your tutorial 2. You want to go through the tutorial 2? Can I ask you all the questions? Huh? So, what question do you have? What questions is bothering you? Yeah, tell me, what question is bothering you?
Sir, you are mainly working on the signaling pathways, isn't it? But there is another strategy against cancer is inhibition of certain enzymes.
Inhibition of?
Enzymes which are involved in, for example, cell replication like telomerase, which stops the cell replication so that we can stop the proliferation of cancer. So what about the work or that strategy of targeting certain enzymes which are very necessary for the cancer cell proliferation and even in metastasis also?
Yeah, you can target anything. You can target enzymes, you can target products from the enzyme, substrates you can target. You can target whichever is important to kill cancer cell and stop cancer cells growing. Anything that enzymes also same way.
Some are like inhibitors they are working on, like
so
that inhibit particular what you call enzymes which are involved in cell replication, so that we can stop the replication.
Sure.
Many of them are using, I think they are used as chemotherapy.
Anything novel idea you have, or people are working on a lot of enzyme studies too. But anything novel ideas which actually kills cancer cells is important. It doesn't matter which one, right? So anybody wants to know anything from the other tutorial? I think there are a lot of questions. Or if you have any questions, tell me tomorrow.
Sir, one. How are therapeutic agents typically identified? How can we identify a target?
How can you identify?
A therapeutic target in case of cancer therapy.
Okay. Means that, like for example, SARC. That's what I've given you a lot of information. I told you a lot of websites to go through and a lot of softwares to analyze. And the day that when we started the SARC story, okay, I told you the SARC story. How did I do the SARC, right? I told you about who was working on SARC in my Vanderbilt University, and why did I start SARC, right? I didn't know which one to start with. At that time, there was not many signaling pathways, because in the pancreatic cancer, I had to start somewhere. So I was actually searching all the molecules, which one to start with, right? Then the SARC popped up. The reason is that whether the SARC is highly expressed or not, I didn't know. Okay, so that is the first thing that I did. I looked at the SARC, whether it expressed high or not, because I knew there is a neighbor who is a SARC person. But if anything happens to SARC here in the because those are different time. Today I may not do the same way, right? Because I have a lot of websites. I can analyze a lot of things. I can correlate with my other studies, and I can come up with a lot of ideas. But at that time there was not these information were not there. So this is what I did. I started with a neighbor, who actually had a SARC antibody also, SARC knockout studies I can do, overexpression studies. That is exactly the tools that you need whenever you start anything research, right? You need a tool. But good thing is that SARC was expressed high in pancreatic cancer, in tumor, when compared to normal. So I was fine. At least I thought I will get a small paper out of it, whether it works or not, right? That's how I started. Look where I went into.
Now if we want to start now, for example, we are starting now.
I gave you a lot of
How we can
information.
identify the target?
I've given you a lot of websites to look into, study. I'm not telling you what you should do. You already got everything that, what do you need to do? Okay, you better start working, and so that you have another day if you want to clarify any of your doubt, right? So it's about you. Okay, we only tell you information. I cannot tell you target. I cannot tell you where to start, how you start, but I can give you my story. Okay, you have to think about you, your situation, where you are and at right now, and what you want to do. Okay, you have to figure it out. Okay, you have to search for it. I only tell you where the information is, but you have to search for it. Get the doubt yourself that whether you're searching or not. You have to doubt about yourself every step of your research. Then only you can get an answer. So don't say that you got an awesome data. Immediately you may say that may be not right. Okay, if you come up with awesome data, if you come to me, then I'll ask you another question. Where is the other one? Then you may not be happy. Things like that. So it happens. It's common. So disappointment is very common. So you have to work hard. Then only you can achieve. Okay? So if you think that all the information will be given out to you, that means that you will not be able to achieve, because that is not yours. If it is, if the information, whatever the science is, want to be yours, you have to work hard. You have to figure it out. You have to find out. Okay? So the story of the SARC, story of the P38, how she started. I told you in the morning how she started. That story is enough for you to dig the story. Like, for example, neighbors and the laboratory, what you worked on. If you are not in the pioneer lab, then find one. Join if you are interested in research. Okay? Then you find a good paper. You may publish another paper, right? But if you want to do yourself, it's fine. You have to work hard. More and more. Find a way. Okay? See, 15 years ago and today literature is changed. I just gave you 2022-23 papers. Believe me, if I had those papers at that time, probably my science will be different. Probably I would, I don't know whether I will be able to survive all these patients, but what I'm saying will be different for sure. Because there is a huge set of data there. Believe me, I'm also looking at it every other day. So, because I also get doubt of this drug, that drug, and synergy of all those, I also see the same. I'm giving you the same information to you, so it's easy for you to look at it and find out, right? So hopefully you'll publish a good paper now on. Yeah. Okay, thanks. All good? So we can wind up today. Okay, if you have any more questions from the other tutorial too, please ask me. Okay, tomorrow. So we will complete today. So you have to come, right? Okay. Thank you, everyone.
Good evening, all. I thank Dr. Nagaraj Nagatihalli to have presented a wonderful his works here, and I thank all the participants to have participated and have been with us till the day 3, and I thank you all. Thank you.